Hello dear student, my name is Hrishikesh Labhade. I am working as an assistant professor at Srimati Vimlaben Khimji Tejokia, SVKT College, Devrali Camp, Nasik. I welcome you all to this online LMS lecture series. Today we are going to learn about a reaction of carbohydrates. This topic includes in Savitribai Phule Pune University's third year BSc semester 1 course. In last lecture, we study about classification and structure of carbohydrates, as well as we see some important physical properties such as epimers, anomers, and inversion, as well as we learn the difference between reducing and non-reducing sugars and understand the mechanism behind mutarotation. The last lecture we end our session with glycosidic bonds in carbohydrates. If you didn't watch that lecture, please go through it. In today's lecture, we are gonna learn about a reaction of glucose with acid, with base, and oxazone formation of glucose with phenylhydrazine, as well as we see the reaction of glucose with oxidizing agents such as bromine water, nitric acid, toluenes and Benedict reagents, as well as we will see the reaction of glucose with reducing agents. We will end our session with biological significance of carbohydrates, the basic functions of carbohydrates in our body. Learning objective. In today's session, student will understand the biological significance of carbohydrates in living organisms. They will learn about reaction of carbohydrates with the glucose as an example. So let's get started. Reaction of glucose with acid. Monosaccharides are generally stable to the action of dilute mineral acid up to 1 normal concentration. But in case of concentrated acid, it decomposes giving dehydrated hydroxymethyl furfural as a product, which is unstable and further decomposed to levulinic acid. The reaction is shown given below. We can see the formation of acid along with carbon dioxide, carbon monoxide and formic acid. Reaction of glucose with base. Glucose react with NaOH undergoes a rearrangement reaction to form mixture of sugars. This mixture is almost optically inactive. This conversion is known as Lowry-de Bruyn-van Enkenstein conversion, which is given in 1895. We can see in the reaction that D-glucose on treatment with dilute NaOH and warming this mixture gives us 2.5% of D-mannose, 63.5% of D-glucose, 31% of D-fructose and 3% are other degraded products. There are many mechanisms suggested by the scientist for this reaction. The reaction takes place via 1,2 enolization according to Awol in 1900. We see that the cis-diol converted into D-mannose. On other hand, the trans-diol gives us D-fructose. The isotopic labeling suggested by isotopic labeling study by Topper in 1951 suggested that two geometrical isomeric enediol form as we see previously. The most accepted mechanism for this is given by Speck in 1958. Enediol did not form in the reaction according to him. The base grabs the alpha H of the D-glucose and gives us intermediate which having a negative charge on the first oxygen of first carbon atom or on the oxygen of second carbon atom, leading to us D-mannose and D-fructose respectively. We can see the mechanism in the image given below. Reaction of glucose with phenylhydrazine. It is one of the most important reaction according to the exam point of view. Glucose reacts with 3 mole of phenylhydrazine to form oxazone. The byproduct of this reaction is aniline and ammonia. There are two mechanisms given to the reaction of glucose with phenylhydrazine. First is a Fischer mechanism. According to Fischer, the first phenylhydrazine molecule react with carbonyl of glucose to give us phenylhydrazone. The second molecule of phenylhydrazine oxidize carbon to OH group to carbonyl group, and it itself reduces to aniline and ammonia, which we see as a side product. At last, the newly formed carbonyl react with a third mole of phenylhydrazine to give us glucazone. We see the mechanism in this image. We can see that a glucose react with first mole to give us phenylhydrazone. The second mole of phenylhydrazine oxidize carbon OH to carbonyl group, and last on reaction with third phenylhydrazine, it gives us glucazone as a final product. There are certain defects in the Fischer theory. First is the phenylhydrazine is strong reducing agent, as we know. Then how can it act as an oxidizing agent in the above reaction? The second question arises that why the reaction stop at C1 and C2 and does not proceed further. These two questions are not answered by the Fischer's theory. Hence, the next theory introduced by Weygand's. The Weygand's mechanism state that the first step involves the formation of phenylhydrazone, which later undergoes Amadori rearrangement. The mechanism of Amadori rearrangement is shown below. Glucose on reaction with phenylhydrazine gives us a cyclic intermediate, which on rearrangement reacting with a further 2 moles of phenylhydrazine to give us glucazone. Now the question arises, what type of intermediate is formed? Blair study by IR UV for the structural elucidation of intermediate, he gets that the third structure shown here are correct and called as a plazazone, which is a yellow solid. Another 1 and 2 are not formed in the reaction. As we see that oxazone formation involve only first 2 carbon atom. Thus, this reaction used to compare the configuration of a symmetric center below C2, that is C3, C4, C5 and so on. For example, D-glucose, D-mannose and D-fructose form the same oxazone. Hence reveals that there is the same exact configuration at a chiral center at carbon number 3, carbon number 5. Now next is a reaction of glucose with oxidizing agent. Oxidation is a process of loss of electron or increase in the oxidation number of any ion, molecule or atom. The reagent which carry out oxidation is known as oxidation agent and itself reduced. Oxidation products of the glucose wholly depend on the nature of reagent used, and these reactions are always used for the structure elucidation of the carbohydrates. First is bromine water oxidation. Bromine is a mild oxidative agent. Bromine water selectively oxidize only aldehyde group to carboxylic acid group, thus converting the aldose into aldonic acid. We can see the image of aldonic acid and aldose reaction in given below. The mechanism behind is that the bromine water specifically oxidize the beta ionomer, giving gamma aldolactone. This compound is further hydrolyzed to an aldonic acid, which undergoes a subsequent ring closure to form a aldanolactone. We can see the mechanism showing the formation of aldanolactone from glucopyranose on reaction with bromine water. Here one important point is that the bromine ion attack anomeric oxygen, and with the loss of HBr from an intermediate, we get the product, but for that the H and HBr should be in a periplanar state, which cannot possible in case of alpha ionomer, and hence alpha ionomers have slow rate of oxidation because the axial H at 3 and 5 hindered the required anti-periplanar arrangement for the oxidation. Nitric acid oxidation. Nitric acid is a strong oxidizing agent. Nitric acid oxidizes both CHU and the terminal CH2OH group to carboxylic acid group of aldoses, forming a dicarboxylic acid on both the end, which is known as aldaric acid. Glucose undergo oxidation using nitric acid to form a glucaric acid. We can see the general reaction as well as the glucose reaction in the image given below. In case of ketosis, reaction with nitric acid give a mixture of acid with a fewer carbon atoms. For example, fructose on oxidation with concentrated nitric acid gives us glycolic acid and tartaric acid. The another oxidation reaction is reaction with lead tetraacetate oxidation. This reagent cleave the carbon-carbon bond having adjacent hydroxyl group attached to them, and they produce the carbonyl compound. We can see the mechanism here. The cyclic intermediate is formed, which upon ring opening gives us a carbonyl compound. If more than two CHOH groups are in continuous fashion, the internal ones are obtained as a formic acid. For example, glycerol on reaction with lead tetraacetate gives us 2 mole of formaldehyde and 1 mole of formic acid. When OH group is adjacent to a carbonyl group, oxidative cleavage takes place. Glyceraldehyde on oxidation with lead tetraacetate gives us a formaldehyde and 2 mole of formic acid. Dihydroxyacetone yields 2 moles of formaldehyde and 1 mole of carbon dioxide. The reaction of this oxidation is shown below. Here is an important point that the periodic oxidation does not cleave compound in which the OH group are separated by CH2 group. Now, the D-glucose on the periodic oxidation yields 1 molecule of formaldehyde and 5 molecules of formic acid. The reaction is shown below. The next is oxidation with a Tollens reagent or Benedict solution. As discussed in the last session, that the reducing sugar reacts with Benedict's and Tollens reagents and give a positive test, that is a silver mirror in case of Tollens reagent and the red PPT in case of Benedict solution. Both reagents gives positive test. The brick red PPT is settled down at the bottom of test tube on treatment with Benedict's reagent, and the silver mirror is formed from inside test tube, showing a silver mirror like finish. Glucose on reaction with Benedict solution gives us sodium salt of gluconic acid plus the red PPT. On the other hand, glucose reaction with Tollens reagents gives us ammonium salt of gluconic acid and a silver mirror. Reaction of glucose with reducing agent. Reduction is a process of gain of electron, and the oxidation numbers are decreases in the process of reduction. The reagent which carries out the reduction is known as a reducing agent. The reducing agent itself gets oxidized during the process. Reducing sugars such as sodium amalgam, high pressure hydrogenation in presence of catalyst, NaBH4, and hydrolytic reduction converts aldoses into their corresponding sugar alcohols. These sugar alcohols are called as alditols. We can see the reaction of D-glucose on hydrogenation. the aldehyde group is converted into alcohol group and the compound is known as D-glucitol. At last, we will end our session with the biological significance of carbohydrates. Carbohydrates supplies and store energy. They provide energy to our body for our day-to-day life and store the energy in fats. Glucose is a sole form of energy for our brain and nervous tissues. Stored glycogen in liver provides energy in absence of glucose. Glucose is a precursor for many organic compounds in our body such as fats, amino acid, coenzyme and nucleic acid. Carbohydrates are also ABO blood makers found on red blood cells. They allow us to distinguish between different types of blood. Immunopolysaccharide plays important resistance against infection in our body and acts like a shield. Carbohydrates present in genetic material of our body. The mucopolysaccharide heparin is an anticoagulant, means it prevents blood clot in our body. The carbohydrate high hyaluronic acid serve as a lubricant and shock absorbent in our body joints. The carbohydrates are also present in RNA and DNA. The sugar is ribose and deoxy respectively. Glucuronic acid acts as a detoxifying agent, means they clean our body by forming a complex with the toxic substances. Carbohydrates also participate in the structure of cell membrane and cellular functions such as cell growth, adhesion and fertilization. Student assignment number 2. Students should have write a note on oxazone formation with glucose. Explain the oxidation of glucose with various oxidizing agents. Explain the concept of mutarotation. There are some MCQs. What will be the product when the 2 mole of D-talose are completely oxidized by the periodate? Question number 5. What if the amylopectin and cellulose are compared, then both these polysaccharides have common? There are 4 options. Student will have to choose one of them. 6th question is, when D-glucose is treated with Benedict's reagent, what is the product? The 7th reagent is seventh question is, which reagent will oxidize D-sorbose? In summary, as we studied that carbohydrates reacts with acid to give us hydroxymethyl furfural. They are resistant to the dilute acids. Glucose react with NaOH and undergoes rearrangement reaction to form the mixture of sucrose. In case of oxazone formation, glucose react with 3 moles of phenylhydrazine to form oxazone, aniline and ammonia as a byproduct. Later we discuss about bromine water oxidation. The bromine water selectively oxidize aldehyde group to a acid group. Nitric acid, which is a strong oxidizing agent, oxidize both aldehyde as well as terminal alcohol group to carboxylic acid group and gives us a dicarboxylic acid with the carboxylic acid present at the 2 ends of the sugar. Later we discuss about lead tetraacetate oxidation, which cleaves carbon-carbon single bond with with adjacent 2 hydroxyl group present on them and gives us a carbonyl compound with the cyclic intermediate. Reduction of aldosaces gives us alditols. Carbohydrates supplies and store energy and are precursor to many organic compounds. Carbohydrates are present in the genetic material of our body. Thank you.